Okay. Hello, everyone, and welcome to today's webinar. My name is Stefania Cesarano from NEMIS Technologies. I'll be responsible for hosting this webinar today. We wait 1 or 2 minutes to start, but meanwhile, this. Why a NEMIS Academy? During my years of working at food producers, I always cared about networking and having a community to share experiences. I was always wondering if they have the same challenges, the same needs, or just talking to somebody for support, helping through recalls, and knowing that I'm not alone with certain problems. But now first, before we begin with the presentation, this. Please submit your questions in the chat room. I will try to get to all of them on this webinar so that our speaker can answer them during our Q&A session. If I do not get to all of our questions, as we have limited time for sure, the questions and the answers will be submitted to all attendees by email. So now our topic today is afraid of recalls. Environmental monitoring can make you safe. We will hear from Mario Hupfeld, CEO of NEMIS Technologies, an overview of that topic. So now, Mario, are you ready? Why not first talk about yourself just a little bit and then your presentation?
Thank you very much, Stefania. Thank you very much for the very nice introduction and also a very warm welcome from my side to the NEMIS Academy, a project that is very dear to our heart here at NEMIS. And I have the honor to kick the webinar series off. So before I start, 1 or 2 words about myself. So my name is Mario Hupfeld. I'm from Germany originally, but now for more than 10 years already in the beautiful country of Switzerland. Came here for my PhD in food microbiology. So for my background, I'm quite technical. So I come from microbiology and molecular biology, but then had the opportunity together with other very nice people to co-found NEMIS Technologies and developing these fast, rapid screening solutions for environmental monitoring. And this is why it's no coincidence that our topic today, also our first topic, is environmental monitoring. And I will now share with you the slides and take you on a journey of around 30 minutes and hope that afterwards we can also have some nice Q&A discussion. As Stefania mentioned, please put all the questions you might have in the chat, and Stefania will try to sort and come back to them at the end of the talk. So now, Stefania, can you confirm that the slides are shown?
Great.
Okay, perfect. So in this case, let's start with the webinar. As mentioned, we will take a short journey around environmental monitoring, where we stand today and where there's maybe still potential to be seen or to be done for environmental monitoring in food safety. And I wanted to start with a very fundamental question, because one thing you can ask in the very beginning of environmental monitoring is, why are we doing environmental monitoring? We usually test our end products if they are safe. So why there's the extra need of doing an environmental monitoring program? And part of the answer you see here a little bit already in this small cartoon. So if you think about a production batch, the testing volume you have for your end product is a very tiny fraction of your production batch usually. And this is not the only limitation you have inherently with end product testing. One thing that is, of course, if you test your end product and you find something, it's already pretty late. It's actually too late. And what happens afterwards is often a very hasty search on what might be the reasons that something in the end product or in the production batch is being found. And there are a lot of factors at play also where, for example, heterogeneity of food samples make it quite unrepresentative to test, and a positive result will tell you mostly nothing about the root cause, except for some exceptions maybe. So it feels a little bit like, if we are being provocative, that end product testing seems a little bit like a lottery. So in contrast, environmental monitoring is a very systematic approach. So I'm sure that most of you that attend the seminar are aware of the zoning of your factory according to different risk profiles, making test plans, HACCP control plans for each zone, and then trying to identify hygiene hotspots. So if you execute very well on environmental monitoring, it can be an incredibly powerful tool, giving you systematic control of your factory and your hygiene state. So you would think that this should be a standard that is very much enforced and encouraged by authorities and being performed then by the food manufacturers. But many studies have shown that there is still quite a bit of potential what can be done in environmental monitoring, and sometimes it's a little bit neglected. So a Swiss study found in 2022 that, looking at Listeria controls, that 2 out of 3 companies did not meet food law requirements. And the most common issues that they found were in the infrastructure of the factory, but also in production hygiene and control of the production environment, which is in the end environmental monitoring. And of course, that poses a bit the questions like, what are the reasons for this? And what we found when we were talking to different food processing company using our screening products is that often the mindset is still a little bit with the food safety regulations in an outdated state. So you have food safety regulations that focuses quite heavily on end product testing. And this leads in the end to authorities being also a little bit insecure how they deal with positives than if they are found in the environment. And we have seen this a couple of times, that then environmental samples are treated as if they would be end product samples. And of course, that is a little bit counter intuitive to what should be done in like a broad screening, where you really try to find your risk spots. And there's, on top of that, very little guidance on how to do environmental monitoring and how often. And this, in the end, leads to the fact that HACCP plans that are really preventive are not incentivized. And what we have seen is that there is a great difference on the execution level of environmental monitoring, depending on the philosophy of the company, because it is very individual. And to illustrate that, I brought a little table here. So how do sampling volumes differ? And you see here ready-to-eat factory sites of different companies, and you see the Listeria tests that are being performed in the environment per month. And you see on the first side that this is a huge variation of several orders of magnitude. One thing you might say immediately is, okay, but probably they are also hugely different in size. This is true to a certain extent. So what we have seen is in many very small factories that no environmental monitoring for Listeria was in place. However, if you go to very large factories that were building their risk mitigation plans mainly on end product testing, you can also have astonishingly few samples that are being taken in the environment. Whereas if you have a relatively small company that is really pushing environmental monitoring as a risk mitigation tool, you can have quite some sample volumes on a relatively small factory size. So the variation is really huge. And one question that comes from this might be, but how much is enough? So how much sampling do you need to do really to feel safe and make your product safe? And the difficulty is that this is not always easy to answer because, of course, factories are very individual. But we brought here two case examples of companies doing changes in environmental monitoring that really made a difference. And these I want to present to you in the next slides. So we have here a meat factory. Classical sampling plan is just taking 25 samples per month. The location are fixed, so they always take the same locations around the factory. And they had a problem finding Listeria in their end product. But they didn't see anything in their current environmental monitoring. So what happened afterwards is they tried a dynamic sampling plan. So they had 25 samples taken like they used to do before, but then evaluated new sampling spots in partly the same rooms, but together then with a food safety consultant. And indeed, what was found is that in one room, Listeria positives could be identified. The hazard plan was changed, so there were new risk spots identified. And this company then changed to having a monthly sampling plan, but with random samples of variation of samples to really always challenge their sampling plan and looking always for potential positives in different parts of the factory. So this is on a relatively low sampling level. You can take this to one level further if you want to really take advantage of trend analysis of lots of samples being taken over a period of time. This we will see now in the next slide. So here, this is a produce company. They took weekly swab samples also for Listeria. They also knew that there was a Listeria contamination present in the plant. And at the beginning of the period, they really scaled up the Listeria swabbing. So they really swabbed in most different places, which led also to the find of more swab positives. Some risk points were identified and eliminated, and in the end, they used this dynamic sampling, then also again scaling down the swabbing to a certain extent when the problem was under control. And what is very nice about this this trend or this continuous sampling that this company is doing is that they really have now a history of months and even now going into the years of swab sampling in the factory in different waves. So if you have if you find positives, they actually already are more able to identify if that is like a potentially persistent problem or something that from time to time pops up with the raw materials that are coming in or certain a certain natural contamination that is being getting rid of via cleaning. So two companies with different sampling volumes, but a common issue, Listeria. This can be, of course, also true for other pathogens like E. coli and Salmonella. But one interesting question here is, what can companies learn from these kind of changes in environmental monitoring, or what does it mean for your own HACCP control plan? And a couple of common things we wanted to point out. One thing that we found time and again, together also with food safety experience consultant, is really being regular in your sampling is very important. So do not take 100 samples once a month. If you can, take 4 times 25 samples, really to get also different exposure of different employee timetables, production timetables, and so on, because otherwise there might be a too large bias in your data. One thing that we have also seen with companies taking charge of environmental monitoring plans is really define a reaction to potential findings. So we mentioned zoning in the beginning. So depending on where you find your Listeria positive, you might have a different reaction schedule to a positive Listeria finding. Maybe you need to re-clean and retest. Maybe you want directly to send a sample from the production batch in. That really is up to the company to define, but really having this plan that you don't ask yourself, okay, how serious do we take a positive or not, is definitely good practice to be ready ahead of time. And one thing that we have seen here in these 2 examples is also these dynamic sampling volumes. So one thing can be to really introduce random sampling points to really challenge your existing HACCP control plan. But it could be also that if you're relatively new in environmental monitoring or don't have a very extensive program at the moment, that you might want to say, I do a so-called swabathon, which would mean I take now for the next 2 weeks every day 20 samples and really establish a baseline of results. And based on this first set of results, I then go further and define a risk plan that I then carry out and see how it works for my factory. This is all relatively independent of budget, so you can, of course, always sample more or always think about how to extend your program. But even if you don't extend it, having these smart ways of dynamic changes and regular intervention points can already do quite a bit to get more out of your data that you're collecting. We also, of course, when we work with companies, have some things that you probably should not do in a seek and destroy approach. There are quite a few things you could mention, but we just focus today on 2 points. And one thing that is very important and dear to our heart is really do not oversimplify your hotspots. So what we have seen in some companies is a risk plan saying, yeah, I only take samples in the drains. I have 5 drains, and if I find Listeria there, then I look further. This is quite a dangerous approach, because if you think about a production line and the niches that Listeria can find on a production line to really stay and persist, if you find it in the drains, it might be already way too late, or you might even never really find it in the drains. So really taking broad indicators and not singular ones is really paramount to a good environmental monitoring program. And the other thing that is, of course, dear to our heart, because we are developing those environmental monitoring tests, is really test your environment if you test your environment, test your environment. If you test your product, test your product. Do not try to mix it up too much. There is always the temptation to use food residues and try to include this in your environmental monitoring. There are valid concerns for testing raw materials or testing certain smear waters or other process materials, but this can be sent to a lab without any issue. And really doing swabbing and sampling, you really should identify the niches where, for example, biofilm can manifest or Listeria can survive on a very low level. These are the kind of hotspots you want to identify really with your swabbing procedures. In the end, if you do this, the return on investment is sometimes it's hard to quantify. Of course, one important point for such a return of investment to mention is you really need an environmental monitoring program where you find something. You know, if you have a Listeria, a Salmonella, or E. coli program and you never find it, your return on investment will, of course, not be the same because you never really have data to interpret. But if you manage to have regular findings of potential hotspots that you can work on, then also a lot of other things come over time as you collect more data that you can actually then interpret and qualify. For example, different quality in raw materials, where you sometimes might get a higher peak of positives. Earlier spotting of niches that might require maintenance because bacteria can more easily stick there and it's hard to clean. Or also the very simple fact that if you establish your own control and self-monitoring system, that this is also a great tool for employee discipline, as if the results are then shared publicly or company-wide. So this was already for companies or different people that might already know quite a bit about environmental monitoring. Maybe take one step back and quickly have a look at, if I'm new to environmental monitoring or I'm suddenly in a crisis, how do I start, or what would be good points to improve my environmental monitoring plan. And one thing that we found that was very interesting for us, because we started as a complete test provider. So we really developed our technology to have this really easy on-site testing. But what we saw quite frequently is that there was quite a need to talk about, where do I actually sample, like how much, when, what parameters might be the best ones to test. And in this regard, what we now encourage companies to do, if they establish a hazard control plan, or if they just want to update it, do a main update, really get an outside perspective. We try to provide this with our customers, as we have with Stefania, for example, a person of a high expertise in quality management of very different food segments. But there are quite a few consultants that offer those services and can really, if they go once with you through the factory, can really make quite a difference. Because one thing that is also very interesting about if you go with an outside perspective through your factory, there are often sampling points coming up which you would have never considered. And we have seen this also again and again, that there are maybe some points in production lines. There was, for one example, was with a produce company that had a production line going through a wall, and then testing the window frame that was going through the wall was actually a hotspot that could be identified. So there's always benefits of bringing people in, even for a short amount of time, and have a look at what your HACCP plan looks like. Another point, when you're starting or when you're thinking about what else can I do in environmental monitoring, it's always to think about the parameters you want to test for. You will usually have hygiene indicators. There's also a question, is ATP enough for cleaning verification, for example, or do you need maybe a total viable count of bacteria that give you a better meaning of the results? Or then if you look at your main pathogen risk, for example, if you are in an industry or a segment where you have pathogenic E. coli potentially in your products, then screening for E. coli in the environment or using an E. coli test also to control for hand hygiene of your employees can be actually quite valuable. Whereas on the other hand, if you have some natural raw materials that are maybe contaminated with pathogens at a relatively low level, but you know this will regularly happen. So for example, raw milk and Listeria monocytogenes, a direct Listeria monocytogenes might be the better choice than a pathogen indicator. So that always depends on your situation and on your also sampling history and state of your factory hygiene on what makes most sense. Coming towards the end, one thing is to work with experts directly for the HACCP control plan. The other thing is if you have established a first line of defense in your factory. So, for example, taking these nice on-site tools that you see here in the pictures and really testing in your factory, have these fast validation cycles. What is always good is to have a backup plan with an accredited laboratory where you can send in samples, either for confirmation or for follow-up analysis. So we had it with quite a few companies that we are working with, that they sent in positive Listeria samples for whole genome sequencing to then check if the positives are new ones or a strain that could maybe persist in the factory and gives you additional information then about how well your hygiene programs are actually working. So we see there is a strong way of actually combining expertise in different ways. Coming there to the end, what we see as a future vision for environmental monitoring is really bringing different aspects together. It's really not only about having the rapid sampling tool or having a perfect expert. It is really a combination of several factors that can really make you successful in really devising the best HACCP control plans that can there be. We do see the need for flexible on-site tools, just also to react very fast and also have maybe continuous cycles of self-improvement at a very fast level whenever needed. We do see that in the future, the self-data collection will be more and more automized, at least from a result perspective. So that means that in the future, companies might be able to get automated trend analysis and already getting some advice on where to sample next or where maybe some weak spots are in the monitoring plan. This combined then with expert knowledge, there are quite a few experts out there that are outbreak consultants and can really help you with specific equipment that you might have in your segment. There are some experts that have a really good knowledge about that. And in the end, if you combine all of that with an accredited validation pathway, you always can combine it into a nice set of data and reporting that you can defend against authorities and auditors. So take-home message there is really monitoring factory hygiene and can ensure safe products. And product testing is definitely not enough. So you will need a good environmental monitoring program to take care of your factory. And already small habit changes can have a big impact. And don't do it all on your own. Get an outside perspective once in a while to really make your plan rock solid. So with this, I'm at the end. If you're interested in what we do as a company and what kind of products we provide for environmental monitoring, of course, feel free to check our websites and our partners. And for now, I'm very happy that you all attended and looking forward to your questions.
So Mario, thank you very much for your presentation. There is really there is a question. We are just going to the session. First question is from Philip. Hi, Philip. Can NEMIS help food companies to improve their environmental monitoring?
Yes, we can do that, of course. As mentioned, we started as a test provider only. So in the beginning, we were only selling tests for detecting Listeria and Salmonella and E. coli. Now we build more expertise over the first years in the market, and we try to help companies actually to set up environmental monitoring plans if they start with our solution. So that can be definitely part of the process.
Okay, thank you. And there, let me be a little bit provocative, Mario. I also have some questions. So in simpler terms, when discussing regulations, and you know that I love standards and guidelines, let's break it a little bit down. Is there a specific formula that incorporates key factors such as production type, high risk, batch volume, production time, and so on, to determine outcomes like a monitoring frequency and the quantity of environmental monitoring? I would really like that formula.
Yeah. Yeah, good question. I think that is definitely a little bit the holy grail, that you can come to quantitative conclusions on saying, like, if I look at your factory, I know exactly how much and what you need to do. Unfortunately, this is still a lot empirical, so there will be no single formula where you can calculate how many samples you might need to take, but there are definitely different factors at play. So for one thing, we kind of touched up on in the presentation is, for example, the age of the factory. So some things that where you have maybe more niches and more unhygienic design components that in the end, you either need to fix it or you need to test more to really get your risk under control. And this is with a lot of small and different things about your factory size, about the hygienic state of your factory, where it is kind of an empirical process to build and to continuously improve the hazard plan. So unfortunately, not a rule of thumb or a universal formula, but I think if you keep working on your environmental monitoring plans, you will eventually get there.
Yes, there you're right, Mario. Okay, I have another question from Martin. Can you give a cost estimate for a typical environmental program approach for a small factory?
That's a very good question. Of course, again, it varies even for very small customers, depending on what kind of products they have, what the factory size is in the end. But you have seen maybe in the table that we presented in the PowerPoint slides, that quite commonly you have sample sizes between 20 and 50 samples per month that you're using for environmental monitoring program. From a cost perspective, this then really depends if you send samples to a laboratory, which can be between 20 and 50 euros, depending on the country you're in and the analysis you want to perform. If you use like a screening tool like the one of NEMIS, you're in a single digit cost per test, and you can very easily calculate. So take, for example, 50 times 7. That would be a relatively simple cost calculation for maybe 1 month of Listeria testing.
Okay, thanks. Oh, there is another one, Mario. If a customer, it's from Philip, if a customer is interested in a testing or a use case of NEMIS tests and device, what is the process? How does it look like?
Yes, so that's a very good question. So we have this very easy starter packages. So you can actually contact us or the distributor in the respective country, and we can set up there a demo and a very easy testing procedure for you to start and feel comfortable with the method. So the easiest is really to contact your local partner and ask for a demonstration, and then this will be organized very quickly.
Okay, thanks again. And there's one question more from Anna. Often question from customer, can they use laboratory thermostat incubator instead of the dry block for incubation?
Yeah, that's also a very good question. So maybe for everyone who is not familiar with our solution, because we didn't talk much about the product today, is you incubate the tubes in a heater or heating block, and the question is do you need to use the NEMIS heating block, or could you use maybe a different one? And we clearly have our solution validated with our heating device. In the end, the heating is not the critical thing in a sense that you need absolutely to use our heating device, but we have our detection tablets which have certain temperature sensitivities. So you would need to use an incubator that only incubates the bottom of the tube and the liquid, and not the caps on top of the tube, because there's the chemistry inside, and if you heat up the whole tube, that might change the shelf life of the tube.
Yes, right there. Mario, may I still be a little bit provocative?
Always.
Always, I know, always. Thanks. Okay, you were talking about Swabatone, right?
Yes.
Okay, let's talk about it. When it comes to sampling, what proves more effective, swabbing or utilizing sponges? What is your opinion about that?
Yes. So that's also a good point. If you think about environmental monitoring, you have also classically different options how to sample. You have sponges, you have swabs, and you have also wipes. From our perspective at NEMIS, we are quite agnostic about it. So we do think that different sampling tools have different usages. So we do see in practice in factories that especially a lot of the risk points are these niches and these points that are actually quite hard to access with a larger sampling tool. So in this case, a swab might be a very useful tool to do sampling. That being said, there's also a trend to sample always larger surfaces. And especially for large and open surfaces, sponges can take on more surface with one sample. So in this sense, for us, it's not either or, but it's always a combination of these sampling tools that make sense in the context.
Mm-hmm. Great. And there is also Rainer just asking about it. Do you have any plans for protocol with sponges, for example, used for meat carcasses?
Okay, so one thing is the sponges and the other thing is maybe the carcass application. Yes, actually, if you stay tuned with us, we are at the moment starting the first pilot tests with a large sampling device that we have developed that can be actually also used within our classical NEMIS tube. So you will be able to use this solution even if you have already a NEMIS luminometer or NEMIS incubator. So this is now in the test phase in Q1. And of course, once we have that, there can be application put on top, like for example, trying to use it on meat carcasses.
Wow, that sounds really interesting. Looking forward to it because, you know, I also love sponges. I think we are quite on time right now. If there are, thank you very much, Rainer, to you. If you, I want to do a closing now. I think we are right on time. Many thanks to Mario. Now we learned about food safety practices, how to reduce risk, and how to deal with risk management. We heard about the return of investment and how the future of factory hygiene can look like. A few powerful thoughts to be at the end here in my mind were, as we plan for the future of food safety and environmental monitoring, it's essential to recognize them as the pillar ensuring the safety of our food. By using knowledge, innovation, and collaboration, we aim to build a future where each food meets the highest standards of safety and quality, and our dedication always to stay ahead of potential risk. Thank you for being a part of this journey, the first one, toward a resilient and pathogen-free tomorrow. As we conclude this enlightening discussion on environmental monitoring, let's anticipate our next rendezvous at the 30th of January, so in 2 weeks' time, where we delve into the fascinating realm of recalls. I really look forward to that. Join us in the upcoming webinar with Stephanie Weinand from Recall Infolink about the task 3 ways to take the crisis out of recalls. Thank you for being part of our continuous journey towards excellence in food safety, and see you soon in 2 weeks' time. Bye.
See you, everyone.